Hello, my name is Alanna Mabey, and this is my research project focusing on sodium butyrate's impairment mechanisms in an in vitro model of IBD. Inflammatory bowel disease, or IBD for short, is a chronic inflammatory disease impacting the intestines. The inflammation caused in this disease can lead to other issues like leaky gut syndrome, which is characterized by the damage done to the intestinal barrier integrity. What this means is that the gut lining is weakened and physical separation between colon cells occurs, which increases its permeability. This is caused by the decreased expression of tight junction proteins like claudin 1, as their role is to regulate and decrease permeability. Sodium butyrate, which is the salt form of the microbial metabolite butyrate, has been a topic of discussion for determining how to treat things like leaky gut and IBD. Since it's a naturally occurring end product of fermentation in the gut and known to provide protection to epithelial cells, it has been studied and shown to be a potential therapeutic agent. It is hypothesized that sodium butyrate will repair inflammation-induced barrier damage by decreasing the permeability of the intestinal lining. The process of the work done in this research started by growing and culturing THP and CACO2 cells. The THPs were differentiated into macrophages, then either left as is and collected, which was the unstimulated conditioned media, or stimulated with LPS in order for macrophages to release cytokines, and this was known as the stimulated conditioned media. The conditioned media was then added to the CACO2 cells to determine its cytotoxicity at different time points by performing an MTT assay. A second MTT was done to treat sodium butyrate on the CACOs to determine its cytotoxicity. Then a third MTT was done to determine if sodium butyrate could repair the damage done by stimulated conditioned media through post-treatment. From the results from the last MTT, one sodium butyrate concentration was chosen to treat CACOs for qPCR. After treatment, RNA was extracted from cells and synthesized into cDNA. From this, the effect of conditioned media and sodium butyrate on the expression of the tight junction protein claudin was determined. Looking at the results, all sodium butyrate treatments were sublethal, whereas the CACOs that were exposed to stimulated conditioned media decreased in cell viability after 48 hours. The conditioned media challenge showed no significant difference in cell viability, which meant sodium butyrate's effects on the cells was not determined. Also, the expression of claudin 1 in all treatment groups were unchanged, so further testing is necessary to determine these treatment group effects. Also, an attempt to develop a permeability model using cell strainers, parafilm, and gelatin was done. A diagram of it can be seen at the bottom of my poster, along with how it was assessed afterwards. Both prototypes were determined to be unsuccessful, as the cells were observed when the parafilm was in focus rather than on top of the strainer membrane, but there is a potential for this model to be better developed and potentially work to assess permeability of the CACO cells. The conclusions made from this research include a successful in vitro model of IBD that was developed after stimulated conditioned media exposure for 48 hours, as well as all sodium butyrate treatments being sublethal, which demonstrated its safety and potential therapeutic capabilities. This model is highly variable, and the results of the conditioned media challenge and PCR support this, as the stimulated conditioned media used in this project did not significantly impact cell viability or claudin 1 expression. Although the stimulated conditioned media did not have the impacts as expected, this IBD model can still help further the development of IBD in vitro, which shows great promise for future research. Future research should focus on producing a uniform batch of stimulated conditioned media for all planned tests to reduce the variability of this model. Also, an ELISA should be run to quantify cytokines in the collected conditioned media, reducing the variability once again and supporting the results that would be obtained from qPCR. For sodium butyrate, different treatments should be evaluated, like pre- and co-treatment instead of post-treatment, or something else to study could be treating macrophages with sodium butyrate directly. This IBD model shows potential for important research in the future, and as studies dive further into it, more treatments and possible therapeutic agents will be discovered. Thank you for listening.